Hey there, welcome to our video. Today, we'll be discussing a groundbreaking study published in Nature Metabolism that explores the role of EphB4 in regulating insulin sensitivity and glucose metabolism. So, grab a snack, get comfortable, and let's get started. Liver EphB4 knockout attenuates insulin resistance in mice. To investigate whether EphB4 is necessary for insulin-induced INSR degradation, we designed human EphB4-specific short hairpin RNAs, shRNAs, and conducted stable knockdown of HepG2 cells. In biotin-labeled cell surface protein assay, insulin-induced cell surface INSR reduction, whereas EphB4 knockdown almost completely rescued this effect. These results strongly show that EphB4 is critical for insulin-induced hepatic INSR degradation. Because total EphB4 knockout mice are embryonically lethal and liver tissue plays an important role in glucose homeostasis, we generated EphB4 liver-specific knockout mice, EphB4-LKO, using the CRISPR/Cas9 technique and the CRELOX-P system. As expected, approximately 70% of the EphB4 mRNA was specifically depleted in the livers of EphB4-LKO mice, and hepatic EphB4 protein levels decreased, accompanied by increased levels of the INSR protein in EphB4-LKO mice. Then, we assessed glucose homeostasis and insulin sensitivity in lean chow-fed and HFD-fed EphB4-FL/FL and EphB4-LKO mice. No differences in body weight, glucose tolerance, or insulin tolerance were noted between lean chow-fed mice of different genotypes. In contrast, in the context of HFD feeding or obesity, the primary hepatocytes from EphB4-LKO mice showed a greater response to insulin stimulation by increasing PAC levels, indicating that EphB4 deletion in hepatocytes improves hepatic insulin signaling. This result was further confirmed by performing a gluconeogenesis assay using these primary hepatocytes, in which insulin exerted a stronger suppressive effect on glucagon activity in hepatocytes from EphB4-LKO mice fed in HFD than in those from control mice, EphB4-FL/FL. In vivo experiments showed that EphB4-LKO mice gained the same amount of weight as wild-type mice, but a marked improvement in glucose and insulin tolerance was observed in EphB4-LKO mice with a reduced basal insulin level compared with the controls. To more accurately quantify in vivo insulin activity and to assess the tissue-specific sites of insulin sensitivity, we performed hyperinsulinemic euglycemic clamp studies. The amount of exogenous glucose required to maintain euglycemia, glucose infusion rate, GIR, during the clamp studies was substantially higher in the EphB4-LKO mice than in the EphB4-FL/FL mice. Hepatic glucose production, HGP, and the ability of insulin to suppress HGP, which reflects hepatic insulin sensitivity, were also improved in the EphB4-LKO mice. Finally, the insulin-stimulated glucose disposal rate, ISGDR, which primarily reflects skeletal muscle insulin sensitivity, and the ability of insulin to suppress plasma-free fatty acid, FFA, levels, which reflects adipose tissue insulin sensitivity, were both increased in the EphB4-LKO mice. Taken together, these data demonstrate that the deletion of EphB4 in hepatocytes leads to a marked improvement in systemic insulin sensitivity under conditions of diet-induced obesity, and these effects were observed in all three major insulin target tissues: liver, muscle, and adipose tissue. As obesity or insulin resistance is often associated with tissue inflammation 21, we assessed whether hepatic EphB4 deletion altered these processes. Hepatic CCL2 and IL-6 levels were significantly reduced in the EphB4-LKO mice, extended data figure 7J, and the expression of CCL2, IL-6, TFA and the macrophage marker F4/80 was downregulated in the epididymal white adipose tissue, APWAT, of EphB4-LKO mice, indicating that EphB4 deletion in the liver results in a less inflammatory state in both the liver and APWAT. To evaluate these effects in a genetic diabetic model, we knocked out EphB4 in the livers of DB/DB mice by crossing EphB4-LKO mice with DB/plus mice. Liver weight and the coefficient of liver in WAT in knockout mice were lower than those in control mice, with no change in body weight between groups. Accordingly, glucose tolerance, insulin tolerance, and hyperinsulinemia were all substantially improved. These results indicate that EphB4 knockdown confers a systemic insulin-sensitive phenotype in mice with genetically induced obesity. Consistent with the findings in EphB4-LKO on an HFD, the hepatic deletion of EphB4 in DB/DB mice significantly reduced inflammation in the livers, indicating that inflammation was ameliorated by knocking down EphB4 in the livers of DB/DB mice. Similar to our findings in the EphB4 stable knockdown HepG2 cells, insulin treatment promoted INSAR degradation in a time-dependent manner in the primary hepatocytes from wild-type mice. However, insulin lost its effect on INSAR degradation, and PAC levels remained high in the primary hepatocytes from EphB4 LKO mice. Thus, EphB4 is necessary for insulin to mediate INSAR degradation in the liver. Discussion. The AP2 binding motifs in EphB4 facilitate INSAR endocytosis and lysosomal degradation, reducing the abundance of INSAR on the cell membrane and insulin sensitivity. Together, these findings suggest that EphB4 serves as a co-receptor of INSAR to inhibit insulin signaling and provide a molecular mechanism of insulin-induced hepatic INSAR degradation and systemic insulin resistance. Other proteins have been reported to interact with INSAR and mediate INSAR endocytosis. For example, inceptors in beta cells promoted INSAR degradation. Our data showed that EphB4 deficiency almost completely reversed the effect of insulin on INSAR degradation in hepatocytes, and EphB4 played a critical role in this process. Thus, in a certain cell type, a specific molecule has evolved to desensitize INSAR signaling. Another case is MAD2 in HepG2 cells 18, which also led to INSAR endocytosis. In contrast to our study, the MAD2-IR interaction was not regulated by insulin. Our study clearly shows that insulin reduces INSR protein levels on the cell membrane and that insulin sensitivity decreases during the course of insulin treatment. Consistent with these results, after the intensification of insulin therapy, some patients with diabetes often reach a state in which further increases in the insulin dosage fail to improve glycemic control while increasing the risks of weight gain. Thus, our study sheds light on the mechanism underlying the failure of glucose management in some patients receiving insulin therapy. The current study reveals marked improvements in glucose tolerance and systemic insulin sensitivity in EphB4 LKO mice fed in HFD, DB/DB EphB4 LKO mice, and DB/DB mice treated with EphB4 inhibitors and shRNAs. So, there you have it. If you want to learn more about this groundbreaking research, be sure to check out the full paper. Thanks for watching, and stay curious.
PharmaPal. PharmaPal.